Hello dear viewers, welcome back again in Glittering Biology with your Bio Guru Alok. Here once again I am along with you with the new topics related with the chapter Molecular Basis of Inheritance under the unit 2nd of your book. In the previous lectures we have already discussed about the genetic material like DNA and RNA and the difference between them along with the experimental proof related with the genetic material. Now today we will just try to cover the central dogma whose concept is required over there, reverse transcription whose basic idea is required for your knowledge. Then we will just talk about the enzymes required in the replication of DNA. If possible, then the transcription topic will be covered during this lecture. Let's begin with the first topic. That one is the central dogma in the molecular biology. If we will just talk about the central dogma, it means what? That we just know that there is a flow of information, genetic information in a cell required for continuation of the characters from one generation to another generation. So the flow of genetic information in a cell is called as the central dogma. We know that the original and ultimate genetic information is present along with the DNA, which act as a master copy. And this information in the DNA is written in the form of the specific codes. They are just called as the genetic code, which is required to develop the characters in new offspring. There are several types of proteins, specific kind of proteins are required for synthesizing or expressing this genetic information in the new cell. That already we have discussed that what are the specific type of RNA which are just conveying the message from one cell to another cell, that is specifically known as the mRNA or the messenger RNA. So here what it is, whenever DNA transfer its genetic information to a molecule of RNA, it is called as the transcription. Means from the DNA, if any information is carried by or carried through the mRNA, then the particular process is called as the transcription. Now RNA goes into the cytoplasm and provides the genetic information to the ribosomes. These ribosomes now synthesize specific proteins according to the message given to them by the mRNA. This process means where the ribosome get involved along with the mRNA for synthesis of specific proteins, that one is called as the translation. In translation, what you will just talk about, tRNA serves to bring the amino acid to ribosomes. At ribosomes, these amino acids get linked together by the peptide bonds to form the polypeptide chain or the protein. That you know very well, that specific amino acids whenever get connected with the polypeptide bonds or peptide bonds, then they are just making a polypeptide chain which later on called as a protein. So what it is, the concept of central dogma was given by the FHC Crick, and he proposed a unidirectional flow of information in which the information regarding protein synthesis is ultimately present in the DNA, which is expressed only in the form of specific protein formation where RNA act as a mediator molecule. Here on the page you can just see there is a specific flow chart it is given where DNA is going to transfer information towards the mRNA. It is just known as the transcription as earlier I discussed, and from the mRNA whenever it is going to produce the proteins for a specific cell, then the process is represented as a translation. Later on, in the concept of central dogma, what we can just consider here, that from the DNA to mRNA, mRNA to protein, this is the normal procedure, where there is at the initiation stage, means where the DNA it is given. Sometimes the DNA forms from the RNA, and that is considered as a reverse transcription. Means from the RNA, if the DNA is forming itself, then it is just called as the reverse transcription. You can say that in the modern era, we know that there are the specific viruses which are specifically known as the retroviruses, where this reverse transcription process is possible for synthesizing their DNA from their RNA, and it is just known as the reverse transcription. The enzyme required for the particular reverse transcription is RNA dependent DNA polymerase. And hope you all are just knowing about the retrovirus, that is the virus which one is containing RNA as a genetic material. With the help of that reverse transcription, these viruses can duplicate their RNA as well, and here the scientist D. Baltimore and Tamin has suggested a revised version of central dogma. This is all about the central dogma. Now, if we will just talk about details in the transcription of RNA, as it is in the previous transcription, it is just asking about the transcription is in your syllabus. So before that one, we are just going to talk about the special enzymes involved in the process of that DNA replication. That one is important for you all. So what are the specific enzymes which we need to study for the replication? Then there are 5 enzymes which are involved in the DNA replication, that is topoisomerase or DNA gyrase. This is the first enzyme. Second enzyme is the helicase. Third enzyme is the DNA polymerase. Fourth enzyme is the primase, and the fifth enzyme is the ligase. One by one, we need to talk about the features of these enzymes. Have a look of this one. Topoisomerase, what it is? Responsible for removing the coiling tension in the DNA molecule. I think we have already discussed about this enzyme in the previous lecture, but in short I am repeating that content. Second enzyme is helicase, which is an important enzyme used to separate the 2 strands of DNA. After removing the tension, whatever the 2 strands are present in the DNA, they get separated with the help of the enzyme helicase. Then the third enzyme is the DNA polymerase, most important enzyme in the DNA replication, and it is main polymerizing enzyme which polymerizes DNA nucleotide to form the DNA strand. Formation of new strand has been managed by the DNA polymerase, and it is involved in the proofreading activities. In eukaryotes, different types of polymerase may be present over there. Then the next one is the primase. Primase, what it is? It is an enzyme which is used for the synthesis of RNA primers. And what is the importance of the RNA, that you know, and it is how it is required for the synthesizing DNA, that we had already discussed. Then ligase is the last enzyme which can catalyze the formation of the phosphodiester bonds between the adjacent 3' hydroxyl and 5' phosphate. That means between adjacent nucleotides during the proofreading. So that is importance of all enzymes which one is involved in the replication of DNA. Then after that, what it is asking in the DNA replication? DNA polymerase. Okay, then next topic we are just going to talk about the transcription of the RNA. Transcription, as earlier we have discussed a little about the transcription in the previous topic, central dogma. The formation of RNA from the DNA is called as the transcription. First step in the process of protein synthesis. You can just see that where there is a transcription, it is written DNA is going to form the in between the that reverse transcription and DNA, and in between the mRNA it is given the transcription. The formation of RNA from the DNA is called as the transcription. And by this process what we are just going to do, that DNA transcripts provides all the required information for protein synthesis to the mRNA molecule. The mRNA molecule has the perfect complementary base sequence over there. In the prokaryotic cells, this transcription occurs in the cytoplasm, while in eukaryotic cells, this transcription takes place in the nucleus. Underline that one. Transcription in prokaryotic cells occurs into the cytoplasm, and in eukaryotes, it is just occurring into the nucleus. What are the basic points related with the transcription that we need to talk about? The enzymes which is responsible for the transcription is called as the RNA polymerase. It is a DNA dependent enzyme which act on the single DNA template and polymerizes RNA nucleotide to form mRNA. Later you will get the different pics, means a flow chart in which you can just summarize that all content. Then an important feature of this particular process is the presence of the template or the antisense strand, or the presence of the sense strand or the coding strand over there. For this purpose, we need to talk about that one of the DNA strand in the double helix hold the genetic information used for the protein synthesis, means it is the parental strand. This is called as the antisense strand or the template or information strand. Later, the complementary strand, means which will be synthesized later on due to the presence of whatever the nitrogenous bases present on one parental strand. On the basis of that, it will just develop the new strand. The complementary strand that binds to sense strand is called as the sense strand or the coding, and it serves as the template for generating a mRNA molecule. That only template strand is transcribed, not the coding strand. This is the important to note down. Means here what it is, the particular template strand is transcribed, not the coding strand. Template strand delivers a copy of sense strand to the ribosome. So that strand which is transcribed is the antisense or the template strand, and the mRNA which is synthesized holds the information exactly similar to that present in the coding strand. Here it is given about the discovery of RNA synthesis. Who has discovered that one? The mechanism of RNA synthesis was worked out in late 1950 in the American investigators, that is the Gerard Horwitz, Samuel B. Weiss, and the Audrey Stevens for his in vitro experiments. What are the transcription units over there? that we will just discuss now one by one. In that one, it is just talking about the different segments of the RNA. The segments of the DNA template strand which transcribes RNA is called as the transcription unit, and they are just containing three parts: promoter region, structural gene, and the terminator gene, terminator region. That are the three important parts for particular process. So one by one we will just talk about the promoter gene. Promoter, what it is? It is a small DNA sequence which is present upstream. It is recognized by the RNA polymerase enzyme, and it indicates which of the two DNA strand is to be read as the template and signals for the direction of transcription. Particular, the promoter also determines the transcription start site, the first nucleotide that will be transcribed into RNA. That is the main function of the promoter. Then the structural, second one is, if I am just taking the second one is as the terminator as it is in your book, then what it is? Terminator must be in the last. Second one will be the structural gene. What it is? RNA coding region. Here what it is a sequence of DNA nucleotide that is copied into an RNA molecule. It actually contains the information for proteins to be synthesized. So that is the structural gene. You can say that one is the RNA coding region. It is the another term we can use for the that structural gene. Here you can see that Benzer used the term cistron for such a functional segment of DNA. So, on in the modern concept, gene is considered a unit of function. That is, functional unit is called as the cistron. A unit of recombination, that one is called as the recon, and the unit of mutation, that is called as the muton, or a unit of the regulation, that is called as the operon. Operon will be later we will discuss in detail. The last third part is the terminator region. What it is a sequence of nucleotide that is present downstream, and it signals where transcription is to end, means where the process will be finished. Terminators are usually part of the coding sequence, so transcription stops only after the terminator has been copied into the RNA. That is all over about the transcription unit. What you must know about the particular few terms, whatever is we are using in this one. That first, the strand that has the polarity 3 dash to 5 dash act as a template and is also referred to as a template strand. Means 3 dash to 5 dash in which one is showing this polarity, that one will be just considered as a template strand. It is called antisense strand or non-coding strand. Means here new synthesis will be not observed. The other strand which has the polarity with the 5 dash to 3 dash is called as the non-template or sense or coding strand. However, it does not code for anything, that is not transcribed. For particular this statement, the logic is as the sense or coding strand is that its sequence is same as that of mRNA except thymine at the place of the uracil. You must be note that all the reference point while defining a transcription unit are made with the coding strand, that is from 5 dash to 3 dash in. Later, here it is given about the, we need to talk about the split genes, but before that one like to discuss about the no mechanism, it is not possible I think. So have a look of the structural genes or the cistrons are two types. On the top of your page it is given. Polycistronic genes, the genes that transcribe mRNA which codes for more than one type of polypeptide chains are called as the polycistronic genes, and RNA as a polycistronic mRNA. These are found only in the prokaryotes. While monocistronic genes, these genes transcribe mRNA which code only for one polypeptide, such as the monocistronic genes and monocistronic mRNA are found in the eukaryotes. Means polycistronic genes are just found into the prokaryotes, while the monocistronic genes are found into the eukaryotes. What are the split genes? As the term is indicating that eukaryotes, the message coded in cistron is discontinuous or interrupted. It is split into the several coding units separated by non-coding base sequence. The coding units are called as the exons. Intervening DNA segments are called as the intron, and such genes as interrupted or split genes by Robert and Shaw. The processed or the mature RNA contains only the copies of exon sequence. Intron sequences are spliced out during the RNA processing. Thus, transcription of an interrupted gene produced the primary transcript RNA or the pre-messenger RNA or heterogeneous RNA, that is the hRNA. Underline that one. It may be asked for the as the abbreviation, means you need to expand the particular hnRNA. It is faithful copy of the interrupted gene. The functional RNA is formed by the removal of introns or unwanted base sequence and the rejoining of its exon or essential sequence. This process is called as the RNA splicing. That's it for today. In the next video, we will just talk about the mechanism of transcription in prokaryotes or in any cell. So, thank you everyone, and prepare notes related with this topic with the help of your book.